Dear aspirant, in this particular course of pharmaceutical analysis and instrumentation, we have seen about the analysis and pharmaceutical analysis in general, as well as the analytical laboratory setup, then various analytical techniques including chemical analysis, spectrophotometric analysis, and chromatographic analysis. And each and every technique like chemical or chromatography or spectrophotometry, we have seen briefly about the analytical method development in each and every technique. But still, it's better to know about this analytical method development in detail as a common module, because any kind of method development you are doing, some of the critical steps are common for all the techniques. So, in this particular module, those common information we are going to see in little bit detail. Let us go and see all those information in the upcoming slides. So why a method development or a analytical method development is required for a analytical technique? Analytical method development is required to establish a suitable method to identify the compound as well as the quantify the compound accurately and precisely, which is very important. So you have to establish a suitable method which give you a proper results, accurate and precise results. So the analytical method development is a process of proving that the developed chemical or a spectrophotometry or a chromatographic method is suitable for its intended use in the analysis of samples during the development and manufacturing of the pharmaceutical drug substance or a drug product. Analytical methods are used to analyze the physiochemical properties, purity or potency of drugs and drug products during the development, manufacturing, and stability of the drug product or drug. And also it is used for the measurement of drugs bioavailability. So in that case, it will be called a bioanalytical method. What are the common types of analytical methods? Identification test, which is generally to do qualitative analysis of chemicals or something. Then physiochemical analysis like optical rotation, refractive index, viscosity, solubility, pH, specific gravity, water content, etc. Then quantitative test for active ingredient in drug substances and drug products. That is generally the assay. Then quantitative test for the impurity content like related substances, process impurities, and degradation impurities during the stability. And then also analyzing the acid neutralizing capacity for the specific drug substances and drug products. And other types of analytical methods include limit test for impurity control like arsenic, lead, etc. For example, chloride content in water. Then elemental analysis or pesticide analysis in the food and agrochemical industries. Then there are certain methods like release test methods like dissolution testing for various solid dosage forms, in vitro release test and in vitro permeation test using Franz diffusion cells for the dosage forms like gel, cream, lotions, ointment, injectable suspensions, etc. Then microbiological testing for oral liquids, soft gelatin capsules, injectables, etc. Then bioavailability testing procedure for the in vivo clinical trials where the bioanalytical method comes into the picture. Then extractable and leachable impurity analysis for packaging as well as finished dosage form materials. Then impurity analysis like nitrosamine analysis. So briefly what we have seen about the different types of analytical methods in the particular slide. Now coming to the steps involved in the analytical method development, first we need to understand the purpose of analytical method why it is required, what is the purpose, what is the expected outcome from the analytical method. For example, if you want to do the assay of the drug or potency of the drug or the drug product, or the objective is to analyze the bioavailability of the drug or the quantity of the drug in a biological matrix, or you want to check the drug stability or the impurity from the drug or drug product, or you want to do the release rate analysis of drug release from the dosage form. So these are the various objectives, but you need to understand the particular objective of the analytical method and the expected outcome. Then the analyte properties, the compound which we are going to measure or analyze, that is called analyte. So the analyte properties needs to be collected like physical chemical properties, storage conditions, light sensitivity, heat sensitivity, solubility, pKa parameters, and then hygroscopicity, lithophilicity, hydrophobicity, etc. Crystallinity also need to be understood for the particular compound to you of interest which we are going to analyze. For this, we need to review the literatures, pharmacopoeias, and other relevant documents for the method development. A tentative analytical method may be chosen from the information obtained from the literature, and this tentative method can be modified or optimized further during the method development. If there is no published method, a tentative method can be tried on trial and error basis based on the information we have got, like physiochemical properties like solubility, affinity constant, pK value, suitability of chromatography like chromophoric nature, etc. can be used for tentative development of a method. The chromophore nature of the molecule is very critical to choose a chromatographic method along with UV absorption or colorimetric technique. So you can use either colorimetric method or a UV spectrophotometric method for a chromophoric compound, or you can use a chromatographic technique, a HPLC equipped with UV or PDA detector for this particular compound. If the compound is ionic nature, if it is an anion or cation, you can use the ion chromatography with a conductivity detector. Then if the compound is very volatile in nature, you can choose for a gas chromatographic method. In case if you are going for a very sensitive molecular method where you want to analyze compounds in nanogram, picograms level, you can go for a liquid chromatography with the mass spectrophotometer detector or gas chromatography with the mass spectrophotometer detector. So during analytical method development, the properties of the analyte need to be understood for selection of appropriate instrument, including the detectors, for the better method outcome. All the analytical method development steps need to be recorded in the laboratory notebook, and this record will help or lead to further modification of the method to get precise, accurate, robust method. While carrying out the optimization of the analytical method, parameter should be changed or can be changed individually, but one at a time preferably, depending on the method outcome. Each and every time you have to check the results and go for further modification. Then the optimization of an analytical method is done in reference to a systematic and procedural plan while making sure to critical follow-up on all the documented steps has been carried out. So when you do the optimization, it should be a systematic and procedural plan, and the documented steps you are following properly so that you will get a appropriate and better results each and every time you optimize the parameters. In a analytical method development or for a analytical method, quantification limit, detection limit, linearity range, analysis time frame, how much time is taken for a particular sample analysis or runtime, then instrument operational cost, which include the solvent cost, buffer cost, column cost, standards, reagent cost, etc., and the sample preparation cost, like whether you involve any extraction technique or cartridges you are using, reagents you are using, those costs should be considered for the merit of the particular analytical method. Then the sample preparation technique, which is very important for the recovery process of the particular analyte from the matrix. And finally, the method should be precise, accurate, robust, and stable to produce the result what we expect from the method. And at the end of the method development, you will have a standard testing procedure which can be verified for different parameters to confirm the method. So generally the standard testing procedure is prepared after the method development and it can be confirmed. The draft can be confirmed after the method verification process. As we have seen, after the method development, we have to perform the method verification to confirm the method. The method verification parameters, which includes system suitability or system precision to confirm the system is or the instrument is working fine, then specificity, then the limit of detection, limit of quantification, linearity or range, accuracy or recovery, then the method precision and intermediate method precision, robustness, stability. So these are the common parameters we have to perform. However, we will see these parameters in detail in the next module, which is called analytical method validation module. Finally, we conclude the method development with a method development report, which containing all the information about the different process we carried out during the method development and the optimization process, which can be referred in the later stage if we have to further modify the method for better results. And also, we conclude with a standard testing procedure, which explain a step-by-step procedure to perform the analysis as per the intended method. So these are the two critical documentation we generally do in the analytical laboratory once the method is developed. And before the validation, these particular documents are generated for the particular method. So aspirants, in this module we have seen the critical steps involved in the analytical method development process and the verification process and finally the documentation generated after the analytical method development. After analytical method development, we generally perform validation wherever it is applicable and the validation how we have to carry out, which we are going to see in the next module. Thank you.